The cell and its function. The cell and its function. The cell and its function. Learning objective. List the function of the major macronutrients in the cell structure and function. List the component of cell and describe their function. Describe the structure of the cell membrane. List the type of a protein on the cell membrane and their function. List the function of a carbohydrate and lipid in the cell membrane. Describe the type and function of a intercellular connection. List different type of a intercellular communication. Describe method that are used to assess cellular function. So glossary of terms. Cell membrane, a lipid bilayer that involves the cell within which proteins are embedded, the so-called fluid mosaic model. Endoplasmic reticulum, a network of tubular and flat vesicular structure in the cytoplasm. The granular endoplasmic reticulum, closely approximated to the ribosome, is involved in protein synthesis. The agranular smooth endoplasmic reticulum helps in lipid synthesis. Golgi body, intracytoplasmic vesicle, involved in intracellular secretion and modification of a protein. Nucleus, the control center of the cell containing large quantities of DNA, which are the genes. These genes determine the characteristic of the cell's protein and also control and promote reproduction of the cell itself. Oxidative phosphorylation, the process by which ATP is synthesized in the mitochondria by the respiratory enzyme. Phagocytosis, ingestion of a large particle. Pinocytosis, ingestion of a minute particles that forms vesicles of extracellular fluid and particulate constituent inside the cell cytoplasm. Now, each of the 100 trillion cell in a human beings is a living structure that can survive for months or year provided its surrounding fluid contain appropriate nutrients. Cells are the building blocks of the body, providing structure for the body's tissue and organ, ingesting nutrients and converting them to energy, and performing specialized function. Cell also contain the body hereditary code that control the substance synthesized by the cell and permit them to take copies of themselves. To understand the function of organs and other structure of the body, it is essential that we first understand the basic organization of the cell and the functions of its component part. Organization of the cell. A typical cell as seen by the light microscope is shown in figure 2.1. Its major part are the nucleus and the cytoplasm. ठीक है. Okay. The nucleus is separated from the nucleus is separated from the cytoplasm by a nuclear membrane, and the cytoplasm is separated from the surrounding by a cell membrane, also called as plasma membrane. The different substances that make up the cells are collectively called protoplasm. Protoplasm is composed mainly of five basic substances: water, electrolyte, protein, lipid, carbohydrate. The principal fluid medium of the cell is water, which is present in most cells except for fat cell in a concentration of 70 to 85%. Important ions in the cell include potassium, magnesium, phosphate, sulfate, bicarbonate, and smaller quantities of sodium chloride and calcium. These all are discussed in more detail in chapter number 6, which consider the interrelation between the intracellular and extracellular fluid proteins. After waters, the most abundant substance in most cells are proteins that normally constitute 10 to 20% of the cell mass. These proteins can be divided into two types: structural protein, functional protein. Structural protein are present in the cell mainly in the form of long filament that are polymer of many individual protein molecules. A prominent use of the such intracellular filament is to form microtubules that provides the cytoskeleton of such cellular organelles as cilia, nerve axon, and the mitotic spindles of the cell undergoing mitosis, and a tangled mass of thin filamentous tubule that holds the part of the cytoplasm and neoplasm together in their respective compartment. Fibrillar proteins fibrillar proteins are found outside the cell, especially in the collagen and elastin fiber of a connective tissue and in the blood vessel walls, tendons, ligaments, and so forth. The functional protein are the entirely different type of a protein and are usually composed of a combination of a few molecules in tubular globular form. These proteins are mainly the enzyme of the cell, and in contrast to the fibrillar protein are often mobile in the cell fluid. Also, many of them are adherent to the membranous structure inside the cell. The enzyme come into direct contact with other substances in cellular fluid and catalyze catalyze specific intracellular chemical reaction. For instance, the chemical reaction that split glucose into its component part into its component part and then combine with glucose to form carbon dioxide and water while spontaneously providing energy for cellular function are all catalyzed by a series of protein enzyme. Then lipids. Lipids are several type of substance that are grouped together because of their common property of being soluble in fat solvent. Especially important lipids are phospholipids and cholesterol, which together constitute only about 2% of the total cell mass. The significance of phospholipid and cholesterol is that they are mainly insoluble in water and therefore are used to form the cell membrane and intracellular membrane barriers that separate the different cell compartments. In addition to phospholipid and cholesterol, some cells contain large quantities of triglyceride, also called a neutral fat. In the fat cell, triglyceride often account for as much as 95% of the cell mass. The fat stored in these cells represent the body's cell mass, the body's main storehouse of energy giving nutrient that can later be used to provide energy wherever is the body it is needed. Carbohydrate. Carbohydrate have little structural function in the cell except as part of a glycoprotein molecules, but they play a major role in nutrition of the cell. Most human cells do not maintain large store of carbohydrate. The amount usually average about 1% of their total mass, but increases to as much as 3% in muscle cell and occasionally 6% in liver cell. However, carbohydrate in the form of dissolved glucose is always present in the surrounding extracellular fluid so that it is readily available to the cell. Also, a small amount of carbohydrate is stored in the cell in the form of glycogen, which is an insoluble polymer of a glucose that can be depolymerized and used rapidly to supply the cell's energy need. Physical structure of the cell. The cell contain highly organized physical structure called as intracellular organelles. The physical nature of each organelle is an importance as the cell chemical constituent for the cell function. For instance, without one of the organelles, the mitochondria, more than 95% of the cell energy released from the nutrients would cease immediately. The most important organelles and other structure of the cell are shown in figure 2.2. Reconstruction of a typical cell showing the internal organelles in the cytoplasm and in the nucleus. Okay, membranous structure of the cell. Most organelles of the cell are covered by a membranes composed primarily of lipid and protein. These membrane include the cell membrane, nuclear membrane, membrane of the endoplasmic reticulum, membrane of the mitochondria, lysosome, Golgi apparatus. The lipid in the membrane provides a barrier that impedes movement of water and water soluble substance from one cell compartment to another because water is not soluble in lipid. However, protein molecule in the membrane often penetrate all the way through the membrane, thus providing specialized pathway often organized into actual pores for passage of a specific substance through the membrane. Also, many other membrane protein are enzymes that catalyze a multitude of different chemical reaction discussed here and in subsequent chapter. Cell membrane. The cell membrane, also called as plasma membrane, involves the cell and is a thin, pliable, इलास्टिक स्ट्रक्चर ओनली 7.5 से लेकर के 10 नैनोमीटर थिक. इट इज कंपोज्ड almost entirely of protein and lipids. The approximate composition is protein जो कि 55%, फॉस्फोलिपिड आपका 25%, कोलेस्ट्रॉल 13%, अदर लिपिड 4%, कार्बोहाइड्रेट आपका 3%. नाउ हिस्टोरिकल विग्निटी. The history of the idea that a combination of a lens could enhance the size of object is difficult to establish in terms of its origins, since different peoples have been ascribed a significant role. Galileo clearly knew of this when constructed his telescope. However, his work has preceded by three spectacle makers of Middelburg, Holland: Hans Janssen, his son Zacharias Janssen, and Hans Lippershey, who developed various lenses in 1590. In 1665, Robert Hooke published his authoritative work Micrographia, where he described the use of his microscope and also provided detailed drawing of many objects. This was a major publication of the Royal Society in Britain and in its time became a bestseller. He was the first to use the word cell. Antony van Leeuwenhoek was a Dutch draper, cloth merchant, who also developed a microscope and even applied them to study the quality of a cloth. He made extensive observation, including that of a microorganism in water, which he called animalcules, little animals. When translated into English and communicated his finding to the Royal Society in a large number of letters, he was finally elected to the Royal Society, a remarkable achievement given that he was not a man of letters but a simple but an extraordinarily curious and inventive merchant. The limitation of light microscopy were overcome with other advances in microscopy. In 1986, the Nobel Prize for Physics was awarded to the Ernst Ruska for his role in the development of electron microscope, which he started working on as a PhD student under Knoll, Max Knoll, who died before the award of the Nobel Prize. Could not be named for the Nobel Prize because Nobel Prize is not awarded posthumously. Okay. Now, the cell membrane lipid barrier impedes penetration by water soluble substances. Figure 2.3 shows the structure of the cell membrane. 2.3 जो कि ये होता है, ठीक है. बस its basic structure is a lipid bilayer, which is a thin double layered film of lipid, each layer only one molecule thick, one molecule thick, that that is continuous over the entire cell surface. In interspersed in this lipid film are longer globular protein. The basic lipid bilayer is composed of three main type of lipid: phospholipid, sphingolipid, and cholesterol. Phospholipid are the most abundant of the cell membrane lipid. One end of each phospholipid molecule is soluble in water, that is, it is hydrophilic. The other end is soluble only in fat, that is, it is hydrophobic. The phosphate end of the phospholipid is hydrophilic and the fatty acid portion is hydrophobic. Because the hydrophobic portion of the phospholipid molecule are repelled by water but are mutually attracted to one another, they have a neutral tendency to attach to one another in the middle of the membrane, as shown in figure 2.3. The hydrophilic phosphate portion then constitute the two surface of the complete cell membrane in contact with intracellular water on the inside of the membrane and extracellular water on the outside of the surfaces. The lipid layer in the middle of the membrane is impermeable to the usual water soluble substances such as ions, glucose, and urea. Conversely, fat soluble substances such as oxygen, carbon dioxide, and alcohol can penetrate this portion of the membrane with ease. Sphingolipid. Jai Shri Ram. Sphingolipid derived from the amino alcohol spigno, sphingosine, sphingosine also have hydrophobic and hydrophilic group and are present in small amount in the cell membrane, especially nerve cell. Complex sphingolipids in the cell membrane are thought to serve several functions, including protection from harmful environmental factor, signal transmission, and as adhesion site for extracellular proteins. The cholesterol molecules in the membrane are also lipid because their steroid nuclei are highly fat soluble. These molecules, in a sense, are dissolved in the bilayer of the membrane. They mainly help determine the degree of permeability the degree of permeability or impermeability of the bilayer to water soluble constituent of the body fluid. Cholesterol control much of the fluidity of the membrane as well. Integral and peripheral cell membrane protein, figure 2.3, also shows globular masses floating in the lipid bilayer. These membrane proteins are mainly glycoprotein. This model of the cell membrane is often referred to as the fluid mosaic model. There are two types of cell membrane protein: integral protein that protrudes all the way through the membrane, and peripheral proteins that are attached only to one surface of the membrane and do not penetrate all the way through. Many of the integral protein provides structural channels or pores through which water molecule or water soluble substance, especially ion, can diffuse between the extracellular and intracellular fluid. These protein channels also have selective properties that allows preferential diffusion of some substance over others. Other integral protein act as a carrier protein for transporting substance that otherwise could not penetrate the lipid bilayer. Sometimes these carrier protein even transport substance in the direction opposite to their electrochemical gradient for diffusion, which is called active transport. Still others act as enzyme. Now, figure 2.3, structure of the cell membrane, showing that it is composed mainly of a lipid bilayer of a phospholipid molecule but with large number of protein molecule protruding through the layer. Also, carbohydrate moieties are attached to the protein molecule on the outside of the membrane and two additional protein molecule on the inside. बस ओके, कितना हो गया? ये तो एक पेज अभी होगा, 2 पेज है यार, खत्म होगा। साला कितना पेज था? 1 2 3 4 5 5 मान के चलते हैं, आहुरी 5टा। बाप रे बाप रे, हे प्रभु, आहुरी 5टा पेज अच्छी। ଏବେ ହେଇଗଲାଣି 28 ମିନିଟ୍। 28 ମିନିଟ୍ ଭିତରେ 2ଟା ପେଜ ମାନେ 4ଟା। Integral membrane protein can also serve as a receptor for water soluble chemicals such as peptide hormone, which do not easily penetrate the cell membrane. Interaction of the cell membrane receptor with specific ligand that bind to the receptor causes conformational changes in the receptor protein. This process in turn enzymatically activates the intracellular part of the protein and induces interaction between the receptor and the protein in the cytoplasm that act as a second messenger, relaying the signal from the extracellular part of the receptor to the interior of the cell. In this way, integral proteins spanning the cell membrane provide a means of conveying information about the environment to the cell interior. Peripheral protein molecules are often attached to the integral protein. These peripheral protein function almost entirely as enzyme or as a controller of a transport of substance through the cell membrane मतलब pores. The function of a protein are summarized in box 2.1 ओके जो कि यहाँ पर है पढ़ेंगे. मेम्ब्रेन कार्बोहाइड्रेट द सेल ग्लाइकोकैलिक्स. Membrane carbohydrate occur almost invariably in combination with protein or lipid in the form of a glycoprotein or glycolipids. In fact, most of the integral protein are glycoprotein and about 1/10 of a membrane lipid molecules are glycolipids. The glyco portion of these molecule almost invariably protrude to the outside of the cell, dangling outward from the cell surface. Many other carbohydrate compounds called proteoglycans, which are mainly carbohydrate substances bound to small protein cores, are loosely attached to outer surface of the cell as well. Thus, the entire outside surface of the cell often has a loose carbohydrate coat called as glycocalyx. The carbohydrate moieties attached to the outer surface of the cell have several important functions. Many of them have a negative electrical charge which gives most cell an overall negative surface charge that repels other negatively charged objects. The glycocalyx of a some cell attaches to the glycocalyx of other cells, thus attaching cells to one another. Many of the carbohydrate act as a receptor substance for binding hormones such as insulin. When bound, this combination activate attached internal protein that in turn activate a cascade of intracellular enzyme. Some carbohydrate moieties enter into the immune reaction as discussed in chapter number 25. Okay. Now box 2.1 function of a protein in the cell membrane एंटीजन हिसाब से काम करेगा. For example, the blood group antigen on the red blood cell membrane. The blood group antigen on the red blood cell membrane. Receptor, for example, for a hormone or neurotransmitter, चैनल के हिसाब से काम करेगा. फॉर वेरियस आयन के लिए ये चैनल बन जाएगा. देन कैरियर प्रोटीन, फॉर एग्जाम्पल फॉर ग्लूकोज एंड अमाइनो एसिड, ये कैरियर प्रोटीन के हिसाब से काम करेगा. पंप के हिसाब से भी काम करता है. फॉर एग्जाम्पल सोडियम पोटेशियम पंप प्रेजेंट ओबीक्यूटसली ऑन ऑल सेल. एंजाइम के हिसाब से काम करेगा. फॉर एग्जाम्पल एडेनिलेट cyclates on the inside of the cell membrane. एंजाइम के हिसाब से काम करा. स्ट्रक्चरल, फॉर एग्जाम्पल स्पेक्ट्रिन इन आरबीसी सेल मेम्ब्रेन. absence of this protein makes the RBC spherical instead of a biconcave disc. Then cytoplasm and its organelles. दीटा पेज पढ़ी सारे ताने चारि पूरा खत्म. साइटोप्लाज्म एंड इट्स ऑर्गेनेल्स. एबे 34 मिनट हो गलानी. The cytoplasm is filled with both minute and large dispersed particles and organelles. The jelly-like fluid portion of the cytoplasm in which the particles are dispersed is called cytosols and contains mainly dissolved protein, electrolyte and glucose. dispersed dispersed in the cytoplasm are natural fat globules, glycogen granules, ribosomes, secretory vesicles, and five especially important organelles: endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, peroxisomes. Now coming to the endoplasmic reticulum. Figure 2.2 कहाँ गया? बस थोड़ा ओवरलैपिंग. Figure 2.2. Yes, everything clear. Figure 2.2 shows a network of a tubular and a flat vesicular structure in the cytoplasm, which is the endoplasmic reticulum. These organelles help process molecules made by the cell and transport them to their specific destination inside or outside the cell. The tubules and vesicles interconnect. Also, their walls are constructed of a lipid bilayer membrane that contain large amount of a protein similar to the cell membrane. The total surface area of this structure in some cell, the liver cell for instance, can be as much as 30 to 40 times the cell membrane area. The detailed structure of a small portion of endoplasmic reticulum is shown in figure 2.4. Okay, nice. The space inside the tubule and the vesicle is filled with endoplasmic matrix, a watery medium that is different from the fluid in the cytosol outside the endoplasmic reticulum. Electron micrograph shows that the space inside the endoplasmic reticulum is connected with the space between two membrane surfaces of the nuclear membrane. Substances formed in some part of the cell enter the space of the endoplasmic reticulum and are then directed to other parts of the cell. Also, the vast surface area of the reticulum and the multiple enzyme system attached to its membrane provide machinery for a major share of the metabolic function of the cell. Ribosomes and the granular endoplasmic reticulums attached to the outer surface of many part of the endoplasmic reticulum are large number of minute granular particle called as ribosome. Where these particles are present, the reticulum is called granular endoplasmic reticulum. The ribosomes are composed of a mixture of RNA and proteins, and they function to synthesize new protein molecules in the cell, as discussed later in this chapter and in chapter number 3. Agranular endoplasmic reticulum, part of endoplasmic reticulum that has no attached ribosome. This part is called the agranular or smooth endoplasmic reticulum. The agranular reticulum function for the synthesis of lipid substances and for other processes of the cells promoted by intrareticular enzyme. Then Golgi apparatus. The Golgi apparatus shown in figure 2.5, a typical Golgi apparatus and its relationship to the endoplasmic reticulum and the nucleus. Golgi apparatus. ओके गोल्गी apparatus ये हो गया. एंडोप्लाज्मिक रेटिकुलम बेसिकली ये हो गया. गोल्गी बेसिकली ये हो गया. एंडोप्लाज्मिक रेटिकुलम ये हो गया. फटाफट चलो. Okay, Golgi apparatus. The Golgi apparatus shown in figure 2.5 is closely related to the endoplasmic reticulum. It has a membrane similar to those of the agranular endoplasmic reticulum. The Golgi apparatus is usually composed of a 4 or more stacked layer of a thin, flat, enclosed vesicle lying near one side of the nucleus. This apparatus is prominent in secretory cell, where it is located on the side of the cell from which the secretory substances are extruded. The Golgi apparatus भगवान. The Golgi apparatus function in association with the endoplasmic reticulum as shown in figure 2.5. Small transport vesicles, also called endoplasmic reticulum vesicle or ER vesicles, continually pinch off from the endoplasmic reticulum and shortly thereafter fuse with the Golgi apparatus. In this way, substance entrapped in the endoplasmic reticulum vesicles are transported from the endoplasmic reticulum to the Golgi apparatus. The transported substance are then processed in the Golgi apparatus to form lysosomes, secretory vesicles, and other cytoplasmic components that are discussed later in this chapter. Lysosomes shown in figure 2.2. लाइसोजोम कहाँ पर है भाई? फिगर 2.2 लाइसोजोम कहाँ 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 किधर है? अच्छा ये are vesicular organelles that forms by breaking off from the Golgi apparatus and then dispersing throughout the cytoplasm. The lysosome provides an intercellular digestive system that allows the cell to digest मतलब number one damaged cellular structure, food particles that have been ingested by the cell, unwanted matter such as bacteria. The lysosome is quite different in various cell types, but it is usually 250 से लेकर के 750 nanometer in diameter. It is surrounded by a typical lipid bilayer membrane and is filled with large number of small granules 5 से लेकर के 8 nanometer in diameter, which are protein aggregates of as many as 40 different hydrolysis digestive enzyme. A hydrolytic enzyme is capable of splitting an organic compound into two or more parts by combining hydrogen from a water molecule with one part of a compound and combining the hydroxyl portion of the water molecule with the other part of the compound. For instance, protein is hydrolyzed to form amino acid. Glycogen is hydrolyzed to form glucose. Uh, Lipids are hydrolyzed to form fatty acid and glycerol. Hydrolytic enzymes are highly concentrated in lysosomes. Ordinarily, the membrane surrounding the lysosome prevents the enclosed hydrolytic enzyme from coming in contact with other substances in the cell and therefore prevent their digestive action. However, some condition of the cell breaks the membranes of some of the lysosomes, allowing release of the digestive enzyme. These enzymes then split the organic substance with which they come in contact into small, highly diffusible substance such as amino acid and glucose. Some of a specific function of lysosome are discussed later in the chapters. Peroxisome. Peroxisomes are similarly physically to lysosome, but they are different in two important ways. First, they are believed to be formed by self replication or perhaps by budding up from the smooth endoplasmic reticulum rather than from the Golgi apparatus. Second, they contain oxidases rather than hydrolases. Several of the oxidases are capable of combining oxygen with hydrogen ion, derived from different intracellular chemicals to form hydrogen peroxide. Hydrogen peroxide is a highly oxidizing substance and is used in association with catalases, another enzyme, oxidase enzyme, present in large quantities in peroxisms. Peroxism to oxidize many substances that might otherwise be poisonous to the cell. For instance, about half of the alcohol a person drink is detoxified into acetaldehyde by the peroxism of the liver cell. In this manner, a major function of peroxism is to catabolize long chain fatty acid. Secretory vesicle. One of the important function of a many cell in secretion is secretion of a special chemical substances. Almost all such secretory substances are formed by the endoplasmic reticulum Golgi apparatus system and are then released from the Golgi apparatus into the cytoplasm in the form of storage vesicle called as secretory vesicle or secretory granules. फिगर 2.6 फिगर 2.6 क्या है अब बाद में देखेंगे फिगर 2.6 shows typical secretory vesicles inside the pancreatic acinar cell. These vesicles stores protein enzyme that are not yet activated. The proenzyme are secreted later through the outer cell membrane into pancreatic duct and hence into the duodenum, where they become activated and perform digestive function on the food in the intestinal tract. Mitochondria. The mitochondria shown in the figure 2.2 and 2.7 are called the powerhouse of the cell. Without them, cell would be unable to extract enough energy from the nutrients and essentially all cellular function would cease. Structure of a mitochondrion modified from D. Robertis. Okay, chalo. Narayana. ଆଉ କେତେଟା ପେଜ୍ ଅଛି 1 2 3 ବଡ଼ ବଡ଼ ପେଜ୍ 3ଟା ଅଛି. Mitochondria are present in all areas of each cell cytoplasm, but the total number per cell varies from less than 100 up to several thousand depending on the amount of energy required by the cell. The cardiac muscle cell, that is cardiomyocytes, for example, use large amount of energy and have far more mitochondria than do fat cell. Than do fat cell मतलब adipocyte, which are much less active and use less energy. Further, the mitochondria are concentrated in those portions of the cell that are responsible for the major share of its energy metabolism. They also vary in size and shape. Some mitochondria are only a few hundred nanometer in diameter and are globular in shape, whereas others are elongated and are as large as 1 micrometer in diameter and 7 micrometer long. Still other are branching and filamentous. The basic structure of a mitochondria shown in figure 2.7 is composed mainly of a two lipid bilayer protein membrane, an outer membrane and a inner membrane. Many infolding of the inner membrane form cells or tubules called cristae onto which oxidative enzymes are attached. The cristae provides a large surface area for chemical reaction to occur. In addition, the inner cavity of the mitochondrion is filled with a matrix that contains large quantity of dissolved enzyme that are necessary for extracting enzyme from nutrients. These enzyme operates in association with oxidative enzyme on the crystals to cause oxidation of the nutrients, thereby forming carbon dioxide and water, and the same time releasing energy. The they liberate they liberate and water. ଏଟା କ'ଣ ଟିକେ 1 minute କ'ଣ ଲେଖିଛି liberated ଓହୋ ହଁ liberated they liberated energy. uh, the liberated energy is used to synthesize a high energy substance called adenosine triphosphate. ATP is then transported out of the mitochondria and diffuses throughout the cell to release its own energy wherever it is intended to performing cellular function. Mitochondria are self-replicating, which means that one mitochondria can form a second one, a third one, and so on, whenever there is a need in the cell for increased amount of ATP. Indeed, the mitochondria contains DNA similar to that found in the cell nucleus. In chapter number 3, we will see that DNA is the basic chemical of the nucleus that control replication of the cell. The DNA of the mitochondrion plays a similar role controlling replication of the mitochondrion. Cells that are faced with increased energy demand, which occur, for example, in skeletal muscle subjected to chronic exercise training may increase the density of mitochondria to supply additional energy required. Cell cytoskeleton, filament, and tubular structure. The cell cytoskeleton is a network of fibrillar protein organized into filament or tubules. These originate as a precursor protein molecule synthesized by ribosomes in the cytoplasms. The precursor molecule then polymerized to form filaments. As an example, large number of actin filaments frequently occur in the outer zone of the cytoplasm called ectoplasm to form an elastic support for the cell membrane. Also in muscle cell, actin and myosin filaments are organized into a special contractile machine that is in the basis for muscle contraction as is discussed in detail in chapter number 14. A special type of stiff filament composed of polymerized tubulin molecules is used in all cell to construct strong tubular structure, the microtubules. Figure 2.8 जो कि ये microtubule teased from the flagellum of a sperm. ओके ठीक है. Figure 2.8 shows the typical microtubule from the flagellum of a sperm. Also, both centrioles and the mitotic spindle of the mitosing cells are composed of a stiff microtubule. Thus, a primary function of a microtubule is to act as a cytoskeleton, providing rapid physical structure, providing rigid physical structure for certain part of the cell. The cytoskeleton of the cell not only determine cell shape but also participate in cell division, allow cells to move, and provides a track-like system that directs the movement of the organelle within the cell nucleus. The nucleus, which is a control center of the cell, send message to the cell to grow and mature, to replicate or to die. Briefly, the nucleus contains large quantities of DNA which comprise the genes. The genes determine the characteristic of the cell's protein, including the structural protein as well as the intracellular enzyme that controls cytoplasmic and nuclear activities. The genes also control and promote reproduction of the cells. The genes first reproduce to create two identical sets of genes, then the cells split by a special process called as mitosis to form two daughter cells, each of which receive one of two sets of the DNA genes. All these activities of the nucleus are considered in detail in chapter number 3. Unfortunately, Unfortunately, the appearance of the nucleus under the microscope does not provide many clues to the mechanism by which the nucleus performs its control activities. Figure 2.9 Figure 2.9 shows the light microscopic appearance of the interphase nucleus during the period between mitosis, revealing the darkly staining chromatin material throughout the nucleoplasms. During mitosis, the chromatin material organizes in the form of highly structured chromosomes, which can be easily identified using the light microscope as illustrated in chapter number 3. Nuclear membrane. The nuclear membrane, also called as nuclear envelope, is actually two separate bilayer membrane, one inside the other. The outer membrane is continuous with the endoplasmic reticulum of the cell cytoplasm, and the space between the two nuclear membrane is also continuous with the space inside the endoplasmic reticulum as shown in figure 2.9. The nuclear membrane is penetrated by several thousand nuclear pores. Large complex of protein molecules are attached at the edge of the pores so that the central area of each pore is only about 9 nanometer in diameter. Even this size is large enough to allow molecules up to 44,000 molecular weight to pass through with reasonable ease. Nuclei and formation of ribosomes. Nucleoli and formation of ribosomes. The nuclei of most cell contain one or more highly staining structure called as nucleoli. The nucleolus, unlike most other organelles discussed here, doesn't have a limiting membrane. Instead, it is simply an accumulation of a large amount of RNA and protein of the type found in the ribosome. The nucleolus becomes considerably enlarged when the cell is actively synthesizing protein. Structure of nucleus. Okay, ये खत्म हो गया. ଆଉ କେତେଟା ପେଜ ବାପରେ ବାପରେ ଆହୁରି 3.5 ପେଜ ପ୍ରଭୁ. The cell and its function ସାରିପାରିବି କି ନା ନାହିଁ କେଜାଣି. ଘଣ୍ଟେ ହେଇଗଲାଣି. Formation of the nucleoli and of the ribosome in the cytoplasm outside the nucleus begins in the nucleus. First, specific DNA genes in the chromosomes cause RNA to be synthesized. Some of this synthesized RNA is stored in the nucleoli, but most of it is transported outward through the nuclear pores into the cytoplasm. Here it is used in conjunction with specific protein to assemble mature ribosomes that play an essential role in forming cytoplasmic protein, as discussed more fully in chapter number 3, functional system of the cell. भाई 1 घंटा हो चुका है. ସାରି ପାରିବେ କି ନା ନାହିଁ ବେ? ଗୋଟେ ଘଣ୍ଟାରେ କେତେଟା ଚାପ୍ଟର ପଢ଼ା ହେଲା, କେତେଟା ପେଜ୍? ବାପରେ, ଗୋଟେ ଘଣ୍ଟାରେ କେତେଟା? 1 2 3 4 ହବ ଆଉ, 4 ହବ. Functional system of the cell. In the remainder of this chapter, we discuss several representative functional system of the cell that make it a living organisms. Ingestion by the cell endocytosis. If a cell is to live and grow and reproduce, it must obtain nutrients and other substances from the surrounding fluids. Most substances pass through the cell membrane by diffusion and active transport. Diffusion involves simple movement through the membrane caused by random motion of the molecules of the substances. Substance move either through the cell membrane pore, or in case of a lipid soluble substance, through the lipid matrix of the membrane. Active transport involve actual carrying of a substance through the membrane by a physical protein structure that penetrate all the way through the membrane. These active transport mechanisms are so important to cell function that are presented in detail in chapter number 4. Very large particle enter the cell by a specialized function of the cell membrane called as endocytosis. The principal form of endocytosis are pinocytosis and phagocytosis. Pinocytosis means ingestion of a minute particle that form vesicle of a extracellular fluid and a particulate constituent inside the cell cytoplasm. Phagocytosis means ingestion of a large particle such as bacteria, whole cell, and a portion of a degenerating tissue. Pinocytosis. Pinocytosis occurs continually in the cell membrane of most cell, but it is especially rapid in some cell. For instance, it occurs so rapidly in macrophages that about 3% of the total macrophage membrane is engulfed in the form of vesicle each minute. Even so, the cytotic vesicles are so small, usually only 100 से लेकर के 200 nanometer in diameter. The most of them can be seen only with an electron microscope. Pinocytosis is the only means by which most large macromolecules such as most protein molecules can enter cell. In fact, the rate at which pinocytotic vesicles form is usually enhanced when such macromolecules attach to the cell membrane. Figure 2.10 demonstrate the successive steps of pinocytosis, showing three molecules of a protein attaching to the membrane. These molecules usually attach to specialized protein receptor on the surface of the membrane that are specific for the type of protein that is to be absorbed. The receptors generally are concentrated in small pits on the outer surface of the cell membrane called coated pits. On the inside of the cell membrane beneath these pits is a latticework of a fibrillar protein called clathrins as well as other proteins, perhaps including contractile filaments of actin and myosin. Once the protein molecule have bound with the receptor, the surface properties of the local membrane change in such a way that the entire pit invaginates inward and the fibrillar protein surrounding the invaginating pit cause its border to close over the attached protein as well as over a small amount of extracellular fluid. Immediately thereafter, the invaginated portion of the membrane breaks away from the surface of the cell, forming a pinocytotic vesicle inside the cytoplasm of the cell. What causes the cell membrane to go through the necessary uh, contortion to form pinocytotic vesicle is still unclear. This process requires energy from within the cell which is supplied by ATP. This process also requires the presence of a calcium ion in the extracellular fluid, which probably react with a contractile protein filament beneath the coated pits to provide the force for pinching the vesicle away from the cell membrane. Phagocytosis. Phagocytosis occur in much the same way as pinocytosis occur, except that it involves large particles rather than molecules. Only certain cells have the capability of phagocytosis, most notable the tissue macrophages and some white blood cells. Phagocytosis is initiated when a particle such as bacterium or a dead cell or tissue debris bind with receptor on the surface of a phagocyte. In case of a bacteria, each bacterium is usually already attached to a specific antibody, and it is the antibody that attaches to the phagocytic receptor, dragging the bacterium along with it. The intermediation of antibodies is called opsonization. Recycling of cell organelle, autophagy. Lysosomes play a key role in the process of autophagy, which literally means to eat oneself. Autophagy is a housekeeping process by which absolute organelles and large protein aggregates are degraded and recycled. Worn out cell organelles are transferred to lysosome by double membrane structure called as autophagosome that are formed in the cytosol. Invagination of a lysosomal membrane and the formation of a vesicle provide another pathway for cytosolic structure to be transported into the lumen of the lysosomes. Once inside the lysosomes, the organelles are digested, the nutrients are reused by the cell. Autophagy contribute to the routine turnover of the cytoplasmic component and is a key mechanism for tissue development. For cell survival, when nutrients are scarce, and for maintaining homeostasis in liver cell, for example, the average mitochondrion normally has a lifespan of only about 10 days before it is destroyed. Clinical box. Pinocytotic and phagocytic foreign substances are digested inside the cell by lysosomes. Almost immediately after a pinocytotic or phagocytotic vesicle appears inside the cell, one or more lysosome becomes attached to the vesicle and empty their acid hydrolysis to the inside of the vesicles as shown in figure 2.11. Thus, a digestive vesicle is formed inside the cell cytoplasm in which the vesicular hydrolysis begin hydrolyzing the protein, carbohydrate, lipids, and other substance in the vesicles. The product of digestion are small molecule of amino acid, glucose, phosphatases, and so forth that can diffuse through diffuse through the membrane of the vesicle into the cytoplasm. What is left what is left of the digestive vesicle called the residual body represents indigestible substances. In most instances, the residual body is finally excreted through the cell membrane by a process called exocytosis, which is essentially the opposite of endocytosis. Thus, pinocytotic and phagocytic vesicle containing lysosomes can be called the digestive organ of the cell. Regression of tissue and autolysis of damaged cell tissue of the body often regress to a smaller size. For instance, this regression occurs in the uterus after pregnancy, in muscles during long period of inactivity, and in mammary gland at the end of lactation. Lysosomes are responsible for much of this regression. Another special role of lysosome is removal of a damaged cell or damaged portion of the cell from the tissue. Damage to the cell caused by heat, cold, trauma, chemical, or any other factor induces lysosome to rupture. The released hydrolyses immediately begin to digest the surrounding organic substances. If the damage is slight, only a portion of the cell is removed and the cell is then repaired. If the damage is severe, entire cell is digested, a process called autolysis. In this way, the cell is completely removed and a new cell of the same type ordinarily is formed by mitotic reproduction of an adjacent cell to take the place of the old one. The lysosome also contain bactericidal agent that can kill the phagocyticide bacteria before they can cause cellular damage. These agents include lysozymes that dissolve the bacterial cell membrane, lysoferrin that binds iron and other substance before they can promote bacteria growth. Acid at pH of about 5.0 that activate the hydrolysis and inactivates bacterial metabolic system. Bravo. Now get the page. History. Synthesis of a cellular structure and endoplasmic reticulum and Golgi apparatus. Specific function of the endoplasmic reticulum. The extensive of the endoplasmic reticulum at the Golgi apparatus in secretory cell has already been emphasized. These These structures are formed primarily on the lipid bilayer membrane similar to the cell membrane, and their walls are located with the protein enzyme that catalyze the synthesis of many substances required by the cell. Most of the synthesis begin in the endoplasmic reticulum. The product form there are then passed on to the Golgi apparatus, where they are further processed before being released into the cytoplasm. First, however, let us note the specific products that are synthesized in the specific portion of the endoplasmic reticulum and the Golgi apparatus. Protein are formed by the granular endoplasmic reticulum. Why? The granular portion of the endoplasmic reticulum is characterized by a large number of ribosomes attached to the outer surface of the endoplasmic reticulum membrane, as discussed in chapter number 3. Protein molecules are synthesized within the structure of the ribosomes. The ribosomes extrude some of the synthesized protein molecules directly into the cytosols, but they also extrude many more through the wall of the endoplasmic reticulum to the interior of the endoplasmic vesicles and tubules into the endoplasmic matrix. Synthesis of a lipid by the smooth endoplasmic reticulum. The endoplasmic reticulum also synthesizes lipid, especially phospholipid and cholesterol. These lipids are rapidly incorporated into the lipid bilayer of the endoplasmic reticulum itself, thus causing the endoplasmic reticulum to grow more extensive. This process occurs mainly in the smooth portion of the endoplasmic reticulum to keep the endoplasmic reticulum from the growing beyond the needs of the cell. Small vesicle called endoplasmic reticulum vesicles or transport vesicle continually break away from the smooth reticulum. Most of these vesicles then migrate rapidly to the Golgi apparatus. Specific function of the Golgi apparatus. Synthetic function of the Golgi apparatus. Although the major function of Golgi apparatus is to provide additional processing of the substance already formed in the endoplasmic reticulum, it also has the capability of synthesizing certain carbohydrate that cannot be formed in the endoplasmic reticulum. This is especially true for the formation of large saccharides, polymer bound with small amount of protein. Important example include hyaluronic acid and chondroitin sulfate. A few of the many functions of hyaluronic acid and chondroitin sulfate in the body are as follows. They are the major component of a proteoglycan secreted in mucus and other glandular secretion. They are the major component of the ground substance or non-fibrous component of the extracellular matrix outside the cell in the interstitial space acting as a filler between collagen fiber and cell. Third, they are principal component of the organic matrix in both cartilages and bone. Fourth, they are important in many cell activities including migration and proliferation, processing of endoplasmic secretion by the Golgi apparatus, formation of a vesicle. Figure 2.1 to summarize the major function of the endoplasmic reticulum and Golgi apparatus. As substances are formed in the endoplasmic reticulum, especially the protein, they are transported through the tubule toward portion of the smooth endoplasmic reticulum that lie nearest the Golgi apparatus. At this point, small transport vesicle composed of small envelope of smooth endoplasmic reticulum continually break away and diffuse to the deepest layer of the Golgi apparatus. Inside these vesicles are synthesized protein and other products from the endoplasmic reticulum. The transport vesically instantly fuse with the Golgi apparatus and empty their contained substances into the vesicular spaces of the Golgi apparatus. Here, additional carbohydrate moieties are added to the secretion. Also, an important function of the Golgi apparatus is to compact the endoplasmic reticular secretion into highly concentrated packets. As the secretion passes towards the outermost layer of the Golgi apparatus, the compaction and processing proceed. Finally, both small and large vesicles continually break away from the Golgi apparatus, carrying with them the compacted secretory substances, and in turn the vesicle diffuses throughout the cell. The following example provide an idea of the timing of these processes. When a glandular cell is bathed in a radioactive amino acid, newly formed radioactive protein molecules can be detected in the granular endoplasmic reticulum within 3 to 5 minutes. Within 20 minutes, newly formed protein are already present in the Golgi apparatus, and within 1 to 2 hours, the protein are secreted from the surface of the cell. Types of vesicle formed by the Golgi apparatus: secretory vesicle and lysosomes. Very huge. ଟିକେ ବ୍ରେକ୍ ନେବା ନହେଲେ ହବନି। ଆଜି କିନ୍ତୁ ସାରିଦେବା। Types of vesicle formed by the Golgi apparatus: secretory vesicle and lysosomes. In a highly secretory cell, the vesicle formed by the Golgi apparatus are mainly secretory vesicle containing protein substances that are to be secreted through the surface of the cell membrane. These secretory vesicles first diffuse to the cell membrane and then fuse with it and empty their substance to the exterior by the mechanism called exocytosis. Exocytosis in most cases is stimulated by the entry of calcium ions into the cell. Calcium ions interact with the vesicular membrane in some way that is not understood and cause its fusion with the cell membrane, followed by exocytosis, that is opening of the membrane outer surface and extrusion of its content outside the cell. Some vesicles, however, are destined for intracellular use. Use of intracellular vesicle to replenish cellular membrane. Some of the intracellular vesicle formed by the Golgi apparatus fuse with the cell membrane or with the membrane of a intracellular structure such as mitochondria and even endoplasmic reticulum. The fusion increases the expanse of these membrane and thereby replenishes the membrane as they are used up. For instance, the cell membrane loses much of its substance every time it forms a phagocytic or pinocytotic vesicle, and the vesicular membrane of the Golgi apparatus continually replenish the membrane. In summary, the membranous system of the endoplasmic reticulum and Golgi apparatus represents a highly metabolic organ capable of forming new intracellular structure as well as secretory substance to be ex- extruded from the cell. The function of the Golgi apparatus are summarized in box 2.2. The mitochondria extract energy from the nutrients. The mitochondria extract energy from the nutrients. The principal substance from which cell extract energy are foodstuff that react chemically with oxygen, carbohydrate, fats, and protein. In the human body, essentially all carbohydrates are converted into glucose by the digestive tract and liver uh, before they reach the other cells of the body. Similarly, proteins are converted into amino acid, fat are converted into fatty acid. Figure 2.13. Okay. Figure 2.13 shows the oxygen and the foodstuff glucose, fatty acid, and amino acid all entering the cell. Inside the cell, the foodstuffs react chemically with oxygen under the influence of enzymes that control the reaction and channel the energy released in proper direction. Briefly, almost all these oxidative reactions occur inside the mitochondria, and the energy that is released is used to form high energy compound ATP. This process is called oxidative phosphorylation. Then ATP, not the original, then ATP, not the original foodstuff, is used throughout the cell to energize almost all the subsequent intracellular metabolic reaction. Functional characteristic of a adenosine triphosphate. Now उसे जानने से पहले box 2.2 function of the Golgi apparatus. Post translational translational processing of proteins, synthesis of glycolipids, synthesis of स्पिंजोमाइलिन, स्पिंजोलिपिड उसे बोला जाता है, प्रोटीन सॉर्टिंग एंड सिक्रेशन. फंक्शनल कैरेक्टरिस्टिक ऑफ एडेनोजाइन ट्राइफॉस्फेट. क्या खत्म कर पाएंगे? लेते हैं एक चैलेंज. साला 2 चैप्टर खत्म करने का रिकॉर्ड मिलेगा। Functional characteristic of a adenosine triphosphate. Okay. Ribose phosphate adenine. Okay, ठीक है। ATP is a nucleotide composed of a nitrogenous base adenine. This one is adenine. The pentose sugar ribose. ठीक है। And 3 phosphate radicals. 3 phosphate radicals. The last 2 phosphate radical are connected with the remainder of the molecule by so-called high energy phosphate bond, which are represented in the formula shown by symbol. Under the physical and chemical condition of the body, each of these high energy bond contain about 12,000 calorie of energy per mole of ATP, which is many times greater than the energy stored in the average chemical bond, thus giving rise to the term high energy bond. Further, high energy phosphate bond is very labile so that it can be split instantly on demand whenever energy is required to promote other intracellular reaction. When ATP releases its energy, a phosphoric acid radical is split away and adenosine diphosphate is formed. This released energy is used to energize many of the cell's other function such as synthesis of substance and muscular contraction. To reconstitute the cellular ATP as it is used up energy derived from the cellular nutrients causes ADP and phosphoric acid to recombine to form new ATP, and the entire process is repeated over and over again. For this reason, ATP has been called the energy currency of the cell because it can be spent and remade continually, having a turnover time of only a few minutes. Chemical processes in the formation of ATP, role of the mitochondria. Upon entry into the cell, glucose is subjected to enzyme in the cytoplasm that convert it into pyruvic acid, a process called as glycolysis. A small amount of ADP is changed into ATP by the energy released during this conversion, but this amount of but this amount accounts for less than 5% of the overall energy metabolism of the cell. About 90% of the cell's ATP formation occurs in mitochondria. The pyruvic acid derived from the carbohydrate, uh, fatty acid from lipids, and amino acid from protein are eventually converted into the compound acetyl coenzyme A in the matrix of the mitochondria. This substance in turn is further dissoluted for the purpose of extracting its energy by another series of enzymes in the mitochondrial matrix, undergoing dissolution in a sequence of a chemical reaction called as citric acid cycle or Krebs cycle. In this citric acid cycle, acetyl coenzyme A split into its component parts, hydrogen atom and carbon dioxide. The carbon dioxide diffuses out of the mitochondria and eventually out of the cell. Finally, it is excreted from the body through the lungs. The hydrogen atom, conversely, are highly reactive and they combine with oxygen that has also diffused into the mitochondria. This combination releases a tremendous amount of energy which is used by the mitochondria to convert large amount of ADP to ATP. The process of these reaction are complex, requiring the participation of many protein enzymes that are integral part of the mitochondrial membranous cells that protrudes into mitochondrial matrix. This the initial event is removal of an electron from the hydrogen atom, thus converting it into a hydrogen ion. The terminal event is combination of a hydrogen ion with oxygen to form water plus release of a tremendous amount of energy to large globular protein protrudes like knobs from the membranes of the mitochondrial cells. This process is called ATP synthesis. Finally, the enzyme ATP synthesis uses the energy from the hydrogen ion to cause conversion of ADP to ATP. The newly formed ATP is transported out of the mitochondria into all part of the cell cytoplasm and nucleoplasm, where its energy is used to energize multiple cell function. This overall process for formation of ATP is called as osmotic mechanism of ATP formation. Use of ATP for cellular function. ଆଉ କେତେଟା ଅଛି ବେ? Energy from the ATP is used to promote three major categories of cellular function. Number one, transport of substance through multiple membrane in the cell, synthesis of a chemical compound throughout the cell, mechanical works. These uses of ATP are illustrated by example in figure 2.14. Number one, to supply energy for the transport of a sodium through the cell membrane to promote protein synthesis by the ribosomes to supply energy needed during muscle contraction. In addition to membrane transport of sodium, energy from the ATP is required for membrane transport of potassium ion. Okay. Potassium ion, calcium ion, and a magnesium ion, phosphate ion, chloride ion, urate ion, hydrogen ion, and many other ion and various organic substances. Membrane transport is so important to cell function that some cell, the renal tubular cell for instance, use as much as 80% of the ATP that they form for this purpose alone. In addition to synthesizing protein, cell makes phospholipids, cholesterol, purines, pyrimidines, and a host of other substances. Synthesis of almost any chemical compound requires energy. For instance, a single protein molecule might be composed of as many as several thousand of amino acid attached to one another by peptide linkages. The formation of each of these linkages requires energy derived from the breakdown of four high energy bonds. Thus, many thousand ATP molecules must release their energy as each protein molecule is formed. Indeed, some cells uses as much as 75% of all the ATP formed in the cell simply to synthesize new chemical compound, especially protein molecule. That is particularly true during the growth phase of the cell. The final major use of ATP is to supply energy for the special cell to perform mechanical work. We see in chapter number 14 that each contraction of a muscle fiber requires expenditure of a tremendous quantities of ATP energy. Other cells perform mechanical work in other ways, especially by ciliary amoeboid motion. The source of energy for all these type of mechanical work is ATP. In summary, ATP is always available to release its energy rapidly and almost explosively whenever in the cell it is needed. To replace the ATP used by the cell, much slower chemical reaction break down carbohydrate, fat, protein and use the energy derived from these process to form a new ATP. More than 95% of this ATP is formed in the mitochondria, which account for the mitochondria being called the powerhouse of the cell. Intercellular connection. How good. In addition to synthesize a protein, cell makes phospholipid, cholesterol, purine, pyrimidine, and a host cell of other substances. Synthesis of almost any chemical compound requires energy. For instance, a single protein molecule might be composed of as many as several thousand of amino acids attached to one another by peptide linkages. The formation of each of these linkages requires energy derived from the breakdown of four high energy bonds. Thus, many thousands of ATP molecules must release their energy as each protein molecule is formed. Indeed, some cells use as much as 75% of all the ATP formed in the cell simply to synthesize new chemical compounds, especially protein molecule. That is particularly true during the growth phase of the cell. The final major use of ATP is to supply energy for special cell to perform mechanical work. We see in chapter number 14 that each contraction of a muscle fiber equal expenditure of a tremendous amount quantities of ATP energy. Other cells perform mechanical work in other ways, especially by ciliary and amoeboid motion. The source of energy for these type of mechanical work is ATP. In summary, ATP is always available to release its energy rapidly and almost explosively whenever in the cell it is needed. To replace the ATP used by the cell, much slower chemical reaction break down carbohydrate, fat, protein and use the energy derived from this process to form new ATP. अरे ये साला पढ़ा था क्या? More than 95% of this ATP is formed in the mitochondria, which account for the mitochondria being called as powerhouse of the cell. I think पढ़ा था. Intercellular connection. While this chapter has primarily dealt with cellular content, it is important to recognize that cell interact with other anatomically and functionally. Anatomic intercellular connection include the following: tight junction, zonula, occludens, occludens mediate adhesion between the cell. They are typically found in the epithelium of the skin, but are also seen elsewhere, such as in the endothelial cell of the blood-brain barrier. Tight junction ensure that membranes are impermeable via the paracellular route. Tight junction are composed of several membrane transmembrane protein including occludin, क्लाउडिन एंड जंक्शनल adhesion molecule जैम जिसे बोला जाता है and are linked to the actin cytoskeleton of the cell. Long thought to be largely responsible for the structural integrity of the cell, tight junctions are now known to be involved with signal transduction into and out of the cell, which modulates cell behavior including cellular proliferation and differentiation. Gap junction located on the lateral surface of the cell link the cytoplasm of the adjacent cell and allow for the movement of ion small molecules and second messengers. Gap junction also contribute low impedance impedance electrical pathway through which electrical activity can be passed from one cell to another. This is the case in the cardiac muscle where the gap junction are an integral part of the uh, cardiac disc. Sorry, integral part of the intercalated disc. Gap junction are formed by protein called as connexin. Six connexin come together to form large structure called as connection with a central pore. The connection is a hemi-channel, and when it align with the connection of adjacent cell, it forms a gap junction. Then adherence junction are also located on the lateral surface of the cell. Unlike the tight junction, they have intermediate permeability. The major protein involved in this junction are cadherin, and various catenin. Like the tight junction, adherence junction are linked to the actin cytoskeleton and are also involved in intracellular signaling and in the regulation of gene expression. Then desmosomes. Desmosomes are located on the lateral surface of the cell in tissue that are particularly subject to mechanical stress such as skin, gastrointestinal mucosa, myocardium, and urinary bladder. The major protein involved in the desmosomes belongs to the cadherin superfamily and include desmoglin and desmocolins. Desmosomes tether the intermediate filament of the cell to the plasma membrane. Like the other intercellular junction, desmosomes have now been shown to have more than a structural function. They are also involved in epithelial proliferation and differentiation. There are various skin diseases which have been associated with the malfunction of desmosomes, including an autoimmune disease called as pemphigus. Pemphigus hemidesmosomes helps to fix the basal epithelial cell to the extracellular matrix. The critical protein involved are the integrin. Hemidesmosomes are critical for cellular differentiation and migration during wound healing. Dysfunction of hemidesmosome result in increased fragility and blistering of the tissue and can be seen pathologically in certain skin conditions such as bullous pemphigoid. Intercellular communication. In the section above, it is clear that some structural intercellular connection also subserve the role of intercellular communication. An example of this is the gap junction. Cell also communicate with each other in other ways. Neuronal communication occurs across a synapse. This is a gap between the neuron where communication occurs by the secretion of a neurotransmitter at the nerve ending. This neurotransmitter crossed the gap मतलब synaptic cleft and act on the receptor on the neuronal membrane of the second neuron. There are a large number of neurotransmitters in the nervous system. Some are excitatory and other are inhibitory. The secretion of one cell might act via receptor located on the adjacent cell and thus influence the function of these adjacent cells. Such as action is called as paracrine. Such an action is called as paracrine. In the islets of Langerhans in pancreas, for instance, there are multiple cells which secrete different hormone. The beta cells secrete insulin, the alpha cells secrete glucagons, and the delta cell secretes somatostatin. Insulin secreted by the beta cell inhibit the alpha cell which secrete glucagons, while somatostatin secreted by the delta cell inhibit both the alpha and beta cell. In contrast, when the secretion of a cell act on the very cell which has secreted it, the action is termed autocrine. Endocrine. Endocrine मतलब intercellular communication act via hormone which are secreted by endocrine gland into the bloodstream to act on the target organ at a distance. In contrast, neurocrine intercellular communication can result from the release of a chemical from the neurons into the bloodstream and acting at a distance from the site of secretion. The action of a posterior pituitary hormone, antidiuretic hormone, and oxytocin are example, and this is discussed in greater detail in chapter number 88. The different types of intercellular communication are illustrated in figure 2.16. Assessing cellular function. In clinical situation, cells within an organ may be affected to the extent that the function of the organ is compromised. In this situation, a patient may present with a history of suggestive of a particular organ involvement. Thus, the presence of abnormal mass or swelling or pain in the particular location direct the physician attention to a particular problem. Sometimes a patient may present with a constellation of symptoms that collectively help localize a problem. Thus, a patient with a low weight loss, intolerance to heat, tremor, anxiety, palpitation, and protruding eyes, expo exothalamus, together with a swelling in the front of the neck might suggest an overactivity of a thyroid gland located in the front of the neck. Clinical examination may add values clues which the physician can objectively elicit. Then there is a variety of laboratory tests that a physician can perform to confirm a diagnosis. ठीक है. There are many ways that the function of cell can be assessed. Biopsy with a microscopy. A biopsy is a technique where tissue is removed from a person. This might be done using a needle or a surgically sampling the tissue. Once the tissue is obtained, it is stained and subjected to microscopy. There are many things that can determine using microscopy. It can determine whether a tissue is overactive or underactive. For instance, when the follicular cell in the thyroid gland which produces thyroxine are overactive, they become longer, that is columnar, while they become flatter if they are not as active, that is cuboidal. There is inflammation or infection of a tissue. Tissue can be acutely inflamed where there will be a larger number of neutrophil and blood cell will be appear congested. Chronic inflammation, on the other hand, will have a preponderance of a lymphocyte and can take on various forms. There is a ischemia or necrosis. Ischemia is an inadequate blood supply to a part or a whole of an organ, while necrosis is the death of the cell due to the inadequate blood supply, disease, or injury. There is a malignancy cancer. Cancer is characterized by an uncontrolled proliferation of the cell. The cancer cell is irregular in size and shape and has a large nucleus with prominent nuclei and scarce cytoplasms. Because cancer cell may have different antigens from normal cell, these abnormal antigen can be detected using a technique called as immunohistochemistry. Here, the antigen is labeled with a specific antibody, and this can be then visualized through a change in color or by tagging the antibody with a fluorophore, which is fluorescent, that is, it re-emits light on light excitation and can therefore be easily seen under a microscope. Immunohistochemistry can also be used to study normal tissue and cell by staining with antibodies attached to the enzyme. When a substrate for enzyme is added to the cell, the enzyme initiates a chemical reaction that causes a color change that can be viewed under microscope. This technique can, for instance, be used to distinguish the slow and fast muscle fiber in the skeletal muscle based on the differences in the enzyme and their concentration in different skeletal muscle fiber. चैप्टर नंबर 16 में इसके बारे में डिटेल में पढ़ेंगे. Evaluation the secretory product of the cell. This is most easily understood in relation to the endocrine hormone producing gland. The function of the gland can be assessed by determining the concentration of the secretion, that is the hormone. Thus high circulating level of thyroid hormone conferring the diagnosis of hyperthyroidism. Although this does not necessarily mean that the problem is in thyroid gland. See chapter number 8-9. Sometimes the absence of a secretory or synthetic product of a tissue or organ may not specifically indicate problem with the tissue. Thus, while albumin, an important plasma protein, is synthesized in the liver, a reduction in plasma albumin could indicate multiple things. It could reflect liver damage, decreased production of albumin, renal damage, increased excretion of albumin, malnutrition, limited availability of the precursor for albumin synthesis. Evaluating circulating cellular content to ascertain cellular damage. When cells in the tissue are damaged or destroyed, they release their cellular product into the circulation. Thus, in pancreatitis and inflammation of the pancreas, pancreatic enzyme such as amylase are seen in higher concentration in blood. Similarly, when the cell of the liver are damaged, as in hepatitis and inflammation of the liver, liver enzymes such as alanine transaminase ALT and aspartate transaminase AST are elevated in the blood. In heart attack, damage to the myocardial cell can be caused by an increased concentration in blood of the content of these cells such as troponin T oblique I. It is important to recognize that those cellular components may not be specific to particular tissue. For instance, creatinine kinase may be released from the damaged myocardial cell as well as from the skeletal muscle. Assessing the electrical activity of the cell. All living cell have a resting membrane potential, जो कि चैप्टर नंबर 7 में पढ़ेंगे. In most cell changes in cell membrane potential precede the biological action of the cell. मतलब contraction, secretion of a hormone or a neurotransmitter, etc. One of the ways to assess cellular function is therefore to evaluate its electrical activity. Hodgkin and Huxley, for instance, recorded the action potential of a single action. Chapter number 9 में पढ़ेंगे. It is now possible to record ionic currents over a small part of the cell membrane as well across a single ion channel using a technique called patch clamp technique. Some cell of the body possesses an intrinsic electrical activity. This can be seen in P, matlab pacemaker cell of the heart in the sinoatrial and atrioventricular node. Chapter number 32. Clinically submitted electrical activity is usually recorded for an organ rather than for a single cell or for a small group of cells. Thus, electrocardiogram ECG records the electrical activity of the heart, chapter number 33 and 34. The electroencephalogram record the electrical activity of the brain, chapter number 122, and electromyogram EMG record the electrical activity of the skeletal muscle. Electromyoneuropathy ENMG uses electrical probes on the surface of the skin to obtain electrophysiological reading from the nerve and muscle cells to determine, among other things, nerve conduction speed. Assessing the genetic function of the cell. Assessing the genetic function of the cell. Karyotyping is a laboratory procedure to evaluate the chromosomes in the cell under light microscopy. Human cells contain 22 pair of autosomal chromosomes and one pair of sex chromosomes. Some of the disorder that can be identified with karyotyping include chromosomal abnormalities that leads to disease in human include Turner syndrome 45X 45XO, Klinefelter's syndrome, the most common male chromosomal disease, otherwise known as 47XXY, has an extra X chromosome, and Down syndrome, a common chromosomal disease caused by trisomy of chromosome 21. The karyotype for Down syndrome is illustrated in figure 2.17. ठीक है, PCR polymerase chain reaction is a method which allows for the amplification of a specific DNA segment from extremely small samples. PCR is used for many things including DNA cloning, for sequencing, functional analysis of the gene, diagnosis of hereditary disease, DNA profiling, and detecting pathogens in infectious disease, among others. Bloods. As you go along in the medical course, you will come across various bloods that are used diagnostic purpose. These are used as an analytical method in molecular biology. The Southern blot, named after the scientist Edwin Southern, aims to detect a specific DNA sequence in DNA samples. The Northern blot accesses gene expression by detecting specific microsomal RNA in a sample. Western blot identifies specific protein in a sample, while Eastern blot evaluate post-translational modification of a protein. There are other blots as well. There are other blots as well. You can thus appreciate that using these various methods, the sequence of a protein synthesis by the genetic machinery of the cell can be assessed. Then biomarkers of a abnormal cell. Biomarker of abnormal cell. A biomarker is a measurable gene or molecule or a characteristic that reflects cellular biology under normal or disease condition. Biomarkers are used for various purposes. They can be used to assess the risk of the disease. This is how likely an individual is to develop a disease. For example, the presence of a mutated BRCA1 and BRCA2 genes indicate an increased risk of breast and ovarian cancer. BRCA, क्या फुल फॉर्म है इसका, देखेंगे. Such individual can be advised to undergo more frequent examination and testing. The prognosis and treatment of a disease, for example, the presence of estrogen receptor in the breast cancer biopsies indicate the individual may respond to the anti-cancer medication that block the effect of estrogen on the cancer cell or reduces the estrogen level in the body response to the treatment. ठीक है, सी में response. Okay, cell culture. Cell biology can also be determined using cell that are grown and maintained under controlled condition. मतलब cell culture. Some cell can be propagated from a fixed number of a division while other can be maintained indefinitely. Not all cell can be used for the cell culture, but the most sustainable cell culture have been from tumor cell from a variety of tissue and organ. A notable example of this is a HeLa, H capital E small l capital A. HeLa cell lines, so named because these cells were derived from the cervical cancer cell of Henrietta Lacks. Henrietta Lacks. Cell culture are grown in a medium that provides nutrients and maintains appropriate physical and chemical conditions such as pH, temperature, and osmotic pressure. Cell culture can be used to study the function of a different type of cell, including metabolic process, cell signaling, and many other experimental studies. Cell culture are also used to make vaccine and to test the effect of toxin and a drug on the cell. Case scenario. A 58 years old man is brought to the emergency department of the hospital. He has been experiencing a crushing type of a chest pain for the last 30 minutes with severe sweating, that is called to diaphoresis. He has a history of a heart attack 2 years earlier and was diagnosed to have diabetes and high blood pressure 10 years before. He has been on irregular treatment for both. The patient is a business executive and is a smoker, one packet a day for the last 25 years. The doctor suspect that he is having an acute heart attack myocardial infarction. They take a blood sample and record his ECG. What test could they perform on the blood sample that will indicate that the cell of the heart are affected? Self-directed learning. Okay. How does an ECG help in assessing the damage to the heart? Self-directed learning. Please note that these will be covered in greater detail in later chapter. At this stage, you need only to read generally to appreciate that various tests can help you access cellular function. ठीक है, फाइनली बहुत खुशी आज मिला है. आई थिंक 2 चैप्टर या कम से कम एक चैप्टर जिसमें अगर हम विचार को करेंगे, बहुत बड़ा चैप्टर था. हालांकि इससे बहुत ज़्यादा भी बच्चे पढ़ते हैं, ऐसा मैं नहीं कर रहा हूँ. बहुत बच्चे तो इससे ज़्यादा भी पढ़ते हैं. 2 3 4 5 6 7 लगभग 8 मान ले तो 16 पेजेस. ऑसम. आज के स्टडी विथ मी ब्लॉग्स में इतना ही. फिर मिलते हैं अगले एक ब्लॉग के साथ. तब तक के लिए जय हिंद, वंदे मातरम. थैंक्स फॉर वॉचिंग.